Hey everybody, my name is Brady, and today my local expert project is going to be over single cell RNA sequencing. So, the best thing that I could use to explain what is single cell RNA sequencing is using the analogy of a fruit smoothie. Say we have a fruit smoothie and we want to explain or investigate what each ingredient does. So, let's say we have blackberries, strawberries, bananas in there. What does each individual ingredient contribute to the fruit smoothie? And also, perhaps, what is each individual characteristic of a strawberry, an orange, or a blueberry in this example? Just going to over a more practical approach to this, imagine we have lung tissue. Inside of lung tissue are fibroblasts, goblet cells, all these other examples of different cells that make up the entirety of the tissue. You have to take each individual cell, sequence its RNA in order to investigate what exactly are the characteristics of a goblet cell or a fibroblast or anything else in that. I would also like to quickly add that this single cell RNA sequencing technique kind of ties into this module where bulk RNA sequencing is sort of like taking a magnifying glass to each cell and being like this one, that one. Single cell RNA sequencing is more like taking a microscope at its highest magnification and being like, yes, it does belong here, but it sort of belongs on this part of the spectrum, if that makes any sense. So, why would we run each individual cell for sequencing instead of just running the entire tissue sample at the same time? Mainly because this gives us an increased definition of the cells being read. Initially, or before single cell RNA sequencing, we have to categorize each individual cell either in this box or that box because that's all we can tell. Meanwhile, if we actually increase the definition of the cells being read, we actually put these cells on a spectrum, where instead, perhaps a lot of them are being expressed over here and over there, and yes, they belong to those respective categories, but maybe they lie somewhere in the middle. That can be very advantageous for us when we're looking at subpopulations within a tissue as well. This can also be used for a drug discovery in personalized medicine. Perhaps an individual does not have the requisite amount of gene expression in certain types of cells, and we can actually give them medicine to help alleviate that. Or perhaps they need to treat their specific types of cells in one population but not the other, and we can give them more targeted medicine. I will say, however, that even though this all sounds great, it's not all oranges and strawberries. Single cell RNA sequencing has a lot of limitations. And the first and foremost, at least in my opinion, is how expensive it can get. Apparently, it costs damn near $5 per cell if you're just running a whole sequence. And you consider how many cells have to make up a tissue, that can stack up immensely. So, hopefully, your pockets align if you're doing this technique, but also it can produce high dimensional data. And I didn't even know how to process that sentence or where even high dimensional data is. More on that later. But finally, it also has these things called dropout events, where essentially, when we're trying to place things on a spectrum, genes that don't have much expression are not even detected. So they don't even register on the spectrum at all. So I guess having a category in that sense would be far more helpful. But going back to high dimensional data, this is just one example of high dimensional data that is resulting from single cell RNA sequencing. And when I saw this, I just thought it was a bunch of dots in a cube. But effectively, what this is that we're so used to seeing data projected in a 2D space. However, because there are so many different categories to categorize a cell on, it effectively adds more dimensions. So they aren't just being registered on a scale from this to this, but also an entirely different dimension. And when you're looking at this, it's very hard for at least like us three-dimensional beings to even comprehend this, which is why we have to use a technique called clustering, where we turn something that looks like this and take all those cells that fit in different categories and turn it into more of a two-dimensional chart like this, where it's much more easy for us to figure this out. Um, I kind of wish there's a better way to describe all these instead of just squares, colors, circles, but at least it's easy on the eye. So if you're going to be running single cell RNA sequencing, you're going to need a couple of controls in order to make sure that your results are accurate in a sense. One positive control that you can use is something called a synthetic spike in RNA, which is effectively like an artificial block of genetic material that we already know the answer to. It's kind of like saying, is 2 plus 2 4 into a computer and making sure the computer does say 4. If it doesn't, they may need to recalibrate your machine. For negative control, it's sort of like running single cell RNA sequencing without any cells to make sure that we don't get any results back, because we're running on an individual cellular level. It's very easy to get contaminants. So if you run this and you do get results, perhaps your pool is contaminated. So we got to go back, make sure it's all clean, make sure we're ready to go. So just a couple figures from the primary manuscripts assigned for this project. I quite like this one a lot, mainly because it actually shows how we're able to separate cells for single cell RNA sequencing. So we take healthy controls and primary Sjorgen's syndrome patients, take samples of their blood, and we run that blood through barcoded gel beads, which effectively takes up each individual cell, runs it through oil to make sure nothing else gets attached to it, and we're able to take that into single cell RNA sequencing and analysis. I'm not sure if you took immunology last semester, but these are kind of giving me some flashbacks. Effectively, based on what chemicals or antibodies they express, we're able to categorize the cells by that. So we have T cells, which don't really exhibit much of anything, versus B cells, which exhibit both high TSN E1 and 2. And from there, we're able to categorize them based on whether or not they're healthy or a control group. So this figure especially shows how defined single cell RNA sequencing can get. This is essentially an analysis of tumoral, normal, and benign tissues in different ovarian cancer patients. But I want to really draw attention to the lines where they separate epithelial, fibroblasts, and leukocytes based on what cells are present. You notice that each individual category has specific colors that are assigned to it. However, you also see that around those lines, you'll see those colors as well. Just drawing your attention right here, leukocytes have a large showing of CD4 T cells, but I also notice that right here there's a overmixture of yellow and also a little bit of purple. So it shows that fibroblasts also have CD4 T cells, which I think is very interesting. And also, we wouldn't be able to do this without single cell RNA sequencing. So, fun fact. So this last figure is a great way to tie directly gene expression to single cell RNA sequencing. So we have right here, this is a study that took different equine tissue samples from adipose tissue, bone marrow, and peripheral blood, and sort of listed from more expressed to less expressed different genes inside each of those tissues. So on the left right here, we have adipose tissue with all of its different associated genes being highly expressed. We're also seeing those same genes expressed in bone marrow and peripheral blood right here. And the same thing for bone marrow. We have a couple right here in adipose tissue, a couple more related ones in peripheral blood, but especially in peripheral blood and bone marrow, lots of the same shared gene expression. But we wouldn't be able to do this without single cell RNA sequencing to see, yes, this cell versus this cell versus this cell. So we'll actually see direct epigenetic heterogeneity, a lot of big words, between different tissue samples, which I always find fascinating. And finally, these are my references that I used to gather information for this presentation. If you have any questions to me about this, please feel free to shoot me a message and I'll try to get back to you. And if it sounds really complicated, I'll find somebody smarter than me to get you a better answer. Thank you for watching, and I hope you have a great rest of your day.